Okay. Hello guys. Good evening everyone. Hope everyone is doing good with your studies. Guys, from today we are going to start a series of videos, series of lectures, especially on the topic of general physiology. Okay. Now today in this class we are going to be mainly concerned with the basics in the physiology, that's the general physiology. Mainly we are going to discuss about cell membrane and associated topics. Okay. Now, if you have any questions, you can all the time ask your questions in the chat box. Okay, I'm just checking my chat box. You can all the time ask your questions. Now, I'm just recording this video especially for the beginners, keeping in mind that you should be able to answer every MCQ which will be coming in your upcoming exams, either it's Fmge, Neet or whatever. Okay, next whatever. So, without any further late, let's start our topic, cell membrane. Okay. Our first topic in general physiology is cell membrane. I hope most of you guys are already good with this topic, but my focus is mainly on those topics which will be coming up in your exams. Having said that, first in cell membrane, let's talk about the composition of cell membrane. Composition of cell membrane. Okay, now we all have millions and millions of cells, and not even millions, we are all having trillions of cells. All the cells are covered by this cell membrane. Now I'm just zooming up that cell membrane. See, this is how the cell membrane looks like. Now, what exactly is this cell membrane? Is it made up of carbohydrates, lipids, proteins, or some other extra thing? See, the composition of cell membrane is such, it's mainly made up of proteins. Okay, the major component, this is a very important MCQ, major component in a cell membrane is protein. After proteins, there are lipids. Okay, don't worry, I will show you lipids and carbohydrates. Okay, so cell membrane is something made up of proteins, lipids, and carbohydrates. If you ask me the ratio, see the percentage, proteins almost account for 50 to 55%, which means in the entire cell membrane, proteins are going to consume how much percentage? 55% is proteins. Lipids how much percent? 40%, and carbohydrate is somewhere between 3 to 5%. So in a cell membrane, this is how the composition is. Major component is proteins, next lipids, and next is the carbohydrates. Okay. After this, you have to know certain exceptions, guys. The clear-cut MCQ is the major component of cell membrane is proteins. But what are the exceptions? Let me write down here exception. Okay. Now the first exception is in Schwann cell membrane. Schwann cell membrane, you will be having highest concentration of lipids. Okay. Now there is a cell called as a Schwann cell. Okay. Now my question for you, if you are really good, okay, if you are really good, just tell me what exactly is this Schwann cell? Can you tell me, guys? Before going to discuss about the protein part or the lipid part, so what exactly is this Schwann cell? Can anyone tell me what is this Schwann cell? Where it exactly presents? See, Schwann cell is something surrounding the peripheral nervous system. Schwann cell surrounds peripheral nervous system. It does myelination. Okay, it causes myelination in the peripheral nervous system. Okay, now just as you guys are very new, see what exactly is peripheral nervous system is. Now we are having something called as a central nervous system. Central nervous system means the brain. See, this is the brain. I'm just showing you the cross section. This is the brain, and down here you will be having a spinal cord. Brain and spinal cord. This is the something called as a central nervous system. You see, CNS. Also remember, from your central nervous system, there are certain nerves coming out. Okay, there are certain nerves coming out. These nerves are called as peripheral nervous system. Simple. The central axial thing, the brain and the spinal cord, brain, brainstem and spinal cord. This is the central nervous system. And the nerves which are coming out of the central nervous system, there are two different types of nerves. The cranial nerves, okay, from the brainstem, the nerves which are coming out, they are called as the cranial nerves, and these the nerves which are coming out of the spinal cord, they are called as the spinal nerves. So cranial nerves and spinal nerves account together as peripheral nervous system. So peripheral nervous system is nothing but cranial nerves as well as the spinal nerves. Now my point is, just I am showing you a one nerve fiber here. Okay, one nerve fiber here. If I cut open that nerve fiber, what you will have inside that nerve fiber? You will be having millions of neurons will be there. Neurons will be there. Okay, just take it as a simple representation of neuron. Now these neurons are myelinated. These neurons are having a insulated sheath. Insulated sheath. Now this myelination, okay, this myelination is done with the help of a cell called as Schwann cell. Okay, so Schwann cells does the myelination of the neuron. Now why we are discussing all this is because, see, this is Schwann cell, it's a cell, right? Now it is also having a membrane. The Schwann cell, it do also have some membrane. Now this Schwann cell membrane has highest concentration of lipids. Okay, normally in a cell membrane you should have highest concentration of proteins, but here the exception is the Schwann cell membrane have highest concentration of lipid. If you ask me, see here in Schwann cell, let me show you here. In Schwann cell, if you see lipids account for almost 80%. 80% is lipids, and proteins are only 20%. Okay, this is the exception. Now after this, let's talk about a few important points which you need to know for your exam. The model which I am showing you here, see, a cell membrane, okay, with all the blue color part. The blue color part is nothing but a lipids. The blue color part is nothing but a lipid. Is it reminding you like a C? The blue color part which I am just labeled is nothing but a lipid which is looking like a C. Now in the C, are you able to appreciate these proteins which are just submerging like icebergs? Okay, so this model which I am showing you, this model was proposed by Singer and Nicholson. The name of this model is called as fluid mosaic model. Okay, so Singer and Nicholson proposed fluid mosaic model. Now, what exactly is this fluid mosaic model? You are having a C. Okay, C represents here lipids in which the icebergs are floating. The icebergs here are nothing but, okay, the icebergs or the mosaic pattern is nothing but icebergs are nothing but the proteins. So which means in the sea of lipids, these proteins are just floating. Okay, so just don't forget for your exam, Singer and Nicholson proposed fluid mosaic model. Now after this, what you should know for your exams. Guys, I'm asking you one question, just try to recollect yourself. In a cell membrane, the highest concentration is what? Proteins, no doubt, sir. Exception is Schwann cell membrane, no doubt. Now other question that can come in your exam is, sir, yes, in a cell membrane, highest concentration is protein. But in which cell membrane the proteins are maximum? Okay, in all our cell membranes, proteins are maximum. But there are certain special cells where the protein concentration is even more than 80%. Okay, now that's the question. The highest concentration of proteins, highest concentration of proteins is seen in which cell membrane? Seen in which cell membrane? Guys, can anyone tell me in the chat box? Can anyone tell me In which cell membrane you will be having highest concentration of proteins? The answer is inner mitochondrial membrane. If you said it, you are excellent. Okay, inner mitochondrial membrane. If you don't find this answer in the MCQs, you can also mark as presynaptic membrane. Okay, so the inner mitochondrial membrane have highest concentration of proteins. Following that, the presynaptic membrane have highest concentration of proteins. You all know, right, what exactly is a presynaptic membrane? Here, this is one nerve, axon with an axonal terminal. Okay, and here is the dendrite of other neuron. So this membrane is called as a presynaptic membrane. This membrane. Now this presynaptic membrane have the highest concentration of proteins after inner mitochondrial membrane. Now after discussing this, now let's start with the each component of the cell membrane. Let's start our discussion first with the lipids. Let's first start our discussion with the topic of lipids. Now we'll go something like this. First we'll discuss about the lipids, all important points which you need to know for your exam. After lipids, we'll discuss about the protein component, carbohydrate component, and we'll go further on. First, the lipids in the cell membrane. Now, the lipids in the cell membrane are mainly classified into three types. Just try to recollect what are the three different types of lipids. The first group of lipids are called as the phospholipids. Phospholipids. Now, in the name itself, it's very clear. Phospholipid means it's a lipid which is attached with a phosphate group. A lipid, see, a lipid which is attached with the phosphate group is called as a phospholipid. There are different types of phospholipids which are present in the cell membrane. It's not, phospholipid is not one single thing. There are different types of phospholipids. Now, second type of lipids are glycolipids. Glycolipids are there in the cell membrane. So what exactly is a glycolipid? It's a carbohydrate, okay, carbohydrate, or I should say something like this. A lipid which is attached to, it's a lipid on which there is a carbohydrate attached. So carbohydrate attached lipid is called as a glycolipid. Okay. Next, third type of lipids are sterols. Okay, the example of sterol is cholesterol. So how many types of lipids are there? Just broadly speaking, how many types of lipids are there in the cell membrane? There are three types of lipids. What are they? Phospholipid, glycolipid, cholesterols. Okay. Now, let's start our discussion with the phospholipids. First, let's start about the phospholipids. Okay. First topic is phospholipids and important points which you need to know for your exam. See, again, as I have already said, there are different types of phospholipids. The first phospholipid I am talking here is phosphatidylcholine. Phosphatidylcholine. Okay. Next phospholipid which I want you to know is sphingomyelin. So phosphatidylcholine. If someone comes to you and asks you, phosphatidylcholine, is it a phospholipid, glycolipid, or cholesterol? It's an example of phospholipid. Sphingomyelin, it's example of phospholipid. Next, phosphatidylserine. Next, phosphatidylinocytol. And last one is cardiolipin. So all what and all I have shown you here. Okay, these are examples of phospholipids. Okay, the lipid attached with the phosphate group. Now let's see all the important points one by one. What you need to know regarding phosphatidylcholine. This phosphatidylcholine is also called as, see guys, the phosphatidylcholine is also called as, very important, dipalmitoyl phosphatidylcholine. Okay, dipalmitoyl phosphatidylcholine, which is also called as the DPPC. So from now on, wherever you see this word DPPC, dipalmitoyl phosphatidylcholine, nothing but our phosphatidylcholine, also known as lecithin. Okay, lecithin. So, what we need to know for our exams regarding this lecithin? Any important point is there? See, this lecithin is a major component. Now, for your exam, this lecithin is a major component of surfactant. Okay. Okay. Now you will get it out. Sir, what exactly is this surfactant? Very simple, guys. Surfactant is something present in the lungs, okay, especially in the alveoli. Okay, in your lungs there are these alveoli, right? Now in your lungs there is a substance present called as a surfactant. Now the surfactant is very important because the surfactant decreases surface tension. For example, if I come to you and by somehow I have taken out the surfactant from your lungs, I have taken out the surfactant from your alveoli, what will happen? Immediately your lungs will collapse because of surface tension in your lungs. In your lungs, there is a destructive force, there is a collapsive force present, which is called as the surface tension. If I take out the surfactant from your lungs are going to collapse. Okay, but your lungs and my lungs are not, our lungs are not getting collapsed. Why? Because our lungs, our alveoli are totally having this surfactant decreases the surfactant, so there is no collapsing of the lungs. Now having said that, so this surfactant, it is made up of a major lipid. Surfactant, it is made up of a major lipid and a minor lipid. Now what is that major lipid? Just now I just discussed with you. The major lipid of surfactant is who? Lecithin. Lecithin is nothing but what? Phosphatidylcholine or DPPC, dipalmityl phosphatidylcholine. Okay, now after discussing about the dipalmityl phosphatidylcholine, now let's talk about sphingomyelin, guys. What you need to know regarding sphingomyelin? See, this sphingomyelin, okay, sphingomyelin is a minor lipid in surfactant. So who is the major lipid? Lecithin. Who is the minor lipid? Sphingomyelin. Okay, so lecithin and sphingomyelin, they forms your surfactant. Lecithin is a major lipid, minor lipid is the sphingomyelin. Okay, now having said that, there is one important MCQ which will keep on coming in the exams, that's the lecithin sphingomyelin ratio. The ratio of lecithin to sphingomyelin. Especially we use this lecithin sphingomyelin ratio to know whether the fetal lungs are matured or not. Okay, normally, see, during pregnancy, think like, try to understand. During pregnancy, where exactly is the baby? Baby is in the uterus. Okay, baby is present inside the uterus, mother's womb. Now, do you think the baby is breathing? Okay, do you think the baby is breathing? No, the baby is not breathing. The baby's lungs are absolutely collapsed. The baby's lungs are not mature. Okay, so during pregnancy and during intrauterine period, the baby's lungs are not matured. Why? Because there is no surfactant. There is no surfactant. Now, if you want to know whether the fetal lungs are matured or not, usually fetal lungs will matured by 34 weeks of period of gestation. Okay, just I want you to know all these points. Fetal lungs are matured by 34 weeks of period of gestation. Usually, the pregnancy term is somewhere around 37 weeks. But by 34 weeks itself, the fetal lungs will start to mature. Why? Why? Because by 34 weeks, the surfactant is getting produced inside the lungs. Now, my question is, how come we know whether the fetal lungs are matured or not, sir? By looking at lecithin and sphingomyelin. Now, this lecithin and sphingomyelin actually will come out of the fetal lungs, will come and dissolve in the amniotic fluid. So, you can collect the amniotic fluid and you can check for lecithin and sphingomyelin. If the lecithin and sphingomyelin, if the ratio of lecithin and sphingomyelin is greater than 2, if the value is greater than 2, it indicates fetal lung maturity. There are other tests also, but at this point I want you to know How fetal lung maturity is assessed by looking at the lecithin sphingomyelin ratio. If the ratio is greater than 2, yes, fetal lungs are matured, sir. But by what time fetal lungs will be matured? 34 weeks of period of gestation. Or if someone comes to you and asks you, usually surfactant production, see, surfactant production starts by, the word is important, surfactant production starts by not 34 weeks, the answer will be 27 weeks. Okay. Now, having said that, okay, let's come back to our general physiology. I just don't go too further into the other topics. Now, we have seen what? We have completed phosphatidylcholine. Also, we have completed sphingomyelin. Two important points are completed. Now, let's talk about phosphatidylserine. Now, just try to recollect, guys. Phosphatidylserine is what? Phosphatidylserine is a lipid in the cell membrane. It's a lipid in the cell membrane. Normally, first of all, let me show you how exactly it looks like. Okay. See, guys, here in this image, please try to concentrate. Phosphatidylserine, here, this area. Phosphatidylserine in a normal cell. The cell number 1 is a normal cell. Phosphatidylserine is actually, it is attached to the cell membrane and it is present where? Intracellularly or extracellularly? Is it expressed intracellularly or extracellularly? It is all the time expressed intracellularly only. It will never be expressed extracellularly. But do you know what happens when the cell is undergoing damage? Okay, when the cell is undergoing damage, now this cell should have to die, right? This cell have to undergo apoptosis. Now at this point, what happens is, see, there will be flipping. There will be flipping of phosphatidylserine from intracellular to extracellular component. There is expression of this phosphatidylserine to outside of the cell. Now, just by looking at this, can we say whether which cells are undergoing apoptosis or not? Yes. So, in those cells which are supposed to undergo apoptosis, we can know by looking at the phosphatidylserine. But important point is, we cannot simply just look the phosphatidylserine with our naked eye. So, what we can do is, you can actually put a dye called as annexin 5. Okay, annexin 5 is the dye. It will go and bind with what? It will go and bind with phosphatidylserine. See, in a normal cell, if you put this phosphatidylserine, it cannot cross the cell membrane. It cannot go and bind to the phosphatidylserine. But in a cell which is undergoing apoptosis, what happens? Now, this annexin 5, it is going and binding to the phosphatidylserine. Whenever it binds with the phosphatidylserine, remember, it glows. It gives fluorescence. Okay, so with this we can clearly say two important points. First thing, phosphatidylserine. First, let me write it here. Phosphatidylserine. Okay, the two important points are, it express always express intracellularly. Okay, now what is the marker of apoptosis? The marker of apoptosis is, the marker means the dye. The marker for apoptosis is annexin 5. So by just putting the annexin 5, you can know which cells are undergoing apoptosis or not. So these are the important points which you need to know regarding phosphatidylserine. After phosphatidylserine, what else do we have? The phosphatidylinositol. Okay, phosphatidylinositol is also a phospholipid which is present in the cell membrane. Now, important points which you should know. Phosphatidylinositol, also called as inositol triphosphate. Okay, phosphatidylinocytol, also called as inositol. All these names are important, guys. Inositol triphosphate. See, just remember this word IP3, phosphatidylinositol, also called as inositol triphosphate. Why? Because whenever we discuss about the proteins, right now we are discussing about the lipids. Whenever we discuss about the proteins, then I will integrate. Okay, so there is a protein which will bind to this phosphatidylinositol called as a lipid anchored protein. Lipid anchored protein, a protein attached to a lipid. Which lipid? Phospholipid. Which phospholipid? Inositol triphosphate. So just for now, don't forget this one word, inositol triphosphate, which is also called as phosphatidylinositol. Now, what else we have to remember for especially from the exam point of view? Now this inocytal triphosphate acts as secondary messenger. Guys, in tomorrow's class or most probably day after tomorrow's class, we will be discussing a topic called as the cellular messengers. Okay, there we will discuss the different types of secondary messengers like cyclic AMP, IP3, DAG. Okay, now just for now, trust me. Now this inocytal triphosphate, it acts as secondary messenger, and whenever you increase inocytal triphosphate levels in the cell, now this inocytal triphosphate IP3, it increases the influx of calcium into the cell, calcium influx. So it increases the calcium influx into the cell. It brings more and more calcium into the cell. Okay, these are the 3 important points which I want you to know. Inocytal triphosphate, also called as IP3, inocytal triphosphate or phosphatidylinositol. It acts as a secondary messenger. It allows the calcium into the cell. Direct question: which phospholipid brings calcium into the cell? IP3, inocytal triphosphate. Okay, inocytal triphosphate also completed. Now the last phospholipid which we have to discuss is a cardiolipin. Now, can you know any cardiolipin? Guys, come on. I want you to answer. Come on, guys. Come on, come on. What are the important points which you have to know regarding cardiolipin? Have you ever heard about anti-cardiolipin antibodies? Cardiolipin. Okay, let me write it down here. Cardiolipin. So, now this cardiolipin, it's a phospholipid, right? Where it's maximally present? It's present maximally in the MCQ. Maximum amount of cardiolipin is present in inner mitochondrial membrane. Okay, it's present maximally in the inner mitochondrial membrane. Okay. Now, what else you have to know? See, for example, if I show you mitochondria, something like this. Okay, there is something called as mitochondria, which are the powerhouses of your cell, where ATP is being produced. Okay, now in this inner mitochondrial membrane, there are certain phospholipids called as cardiolipins. But unfortunately, do you know what happens? There are certain individuals who produce antibodies. Okay, because of autoimmune disorders, there are certain individuals who will produce antibodies against this phospholipid, the cardiolipin, which are called as cardiolipin antibodies, anti-cardiolipin antibodies. My question for you is, in which diseases in your blood you will have anti-cardiolipin antibodies? Anti-cardiolipin antibodies are seen in 3 conditions. At least I want you to answer 2. At least I want you to answer 2. What are they? First one start with the letters S. S. Come on, guys. What is the excellent, Bruce? It is syphilis. Second one is excellent, Apla syndrome. Okay, Apla syndrome. And third one is SLE, systemic lupus erythematosus. Okay, so there are 3 diseases where you will have anti-cardiolipin antibodies. Now here I just want to integrate. Now see, just tell me, syphilis is what kind of disease? Syphilis is what kind of disease? Is it a mitochondrial disorder? What? Is it a fever because of some vector? No. So syphilis is a sexually transmitted disease, and the causative organism, can anyone tell me? Come on, doctors. What is the causative organism? Or what is the causative agent? What is the causative agent of syphilis? Syphilis is caused by, yes, excellent, Bruce. It is, oh my god, someone is saying HPV. No, it is Treponema pallidum. Okay, so Treponema pallidum, it's a spirochete. Okay, the spirochete in the sense they are spiral. These organisms are spiral in structure. So Treponema pallidum is the organism which causes syphilis. Okay. Now Guys, especially for the OBG point of view, you have to know this APLA syndrome. Those, especially that female who have this APLA syndrome, as in the name itself, it's there, anti-phospholipid antibody. What is that phospholipid? The phospholipid is nothing but the cardiolipin. Okay, so in anti-phospholipid antibody syndrome, the antibodies are getting against the cardiolipin. So those females who ever have this anti-phospholipid antibody syndrome, they will have recurrent abortions. Okay, recurrent abortions. So for your entire medical career, the moment you listen the word APLA syndrome, the first thing that comes and strikes your mind is recurrent abortions. That female who have APLA syndrome will be having recurrent abortions. Okay. Now, especially from the OBG point of view, I want you to know this one more point. See, for example, imagine that I am a female who is having APLA syndrome. Now, what will I have? I will have recurrent abortions. I cannot have my pregnancy till to the 37th week. So there is something called as the first trimester, second trimester, and third trimester. Okay, the entire pregnancy was divided into first trimester, second trimester, third trimester. If I am having APLA syndrome, I will have recurrent abortion. But when? In first trimester, second trimester, third trimester. MCQ. In second trimester. Okay, APLA syndrome causes recurrent abortions in the second trimester. That's also important MCQ, which I want you to know. Now, after this, let's discuss about the glycolipids. Guys, see, we have completed the topic of phospholipids. Phospholipids topic is completed. Now let's talk about the glycolipids. Already I have explained to you what exactly is a glycolipid. A lipid attached to the carbohydrate group. Here, there are not much important points which I want you to know, especially the glycolipids component is mainly dealt with the biochemistry. But here in physiology, the important points what I want you to know is there are two important glycolipids. Okay, two important glycolipids. Okay, let me write it down here. Glycolipids are mainly divided into cerebrosides, and gangliosides. Okay, cerebrosides and gangliosides. Cerebroside and ganglioside, direct question. Are the example of phospholipid or glycolipid? They are example of glycolipids. Now here, the repeatedly asked MCQ is this ganglioside. These gangliosides are further classified into GM1, GM2, GM3 different types. Here I want you to know there is a ganglioside called as the Gm1 ganglioside. This is a repeated MCQ. Gm1 ganglioside. Can anyone tell me what is the importance of this Gm1 ganglioside which is present on the cell membrane? This Gm1 ganglioside, it is a glycolipid present on the cell membrane. The importance is, let me show you. See here. Are you able to appreciate this Gm1 ganglioside on the cell membrane? See, this is the cell membrane. Okay, especially in the GIT. On the cell membrane, there is something called as a Gm1 ganglioside. Now, this Gm1 ganglioside acts like a receptor. It's acting like what? It's acting like a receptor. Receptor for what? Guys, it's acting like a receptor for the chlorotoxin. Okay, there is an organism called as Vibrio cholera. Okay, Vibrio cocci, right? Vibrio cholera is there. Now, whenever you got infection, Vibrio cholera, what will happen? Now, this Vibrio cholera, this organism is going to produce a toxin called as a chlorotoxin. Now, this chlorotoxin is, it will bind with this Gm1 ganglioside. Now, whenever it binds with the Gm1 ganglioside, this Gm1 ganglioside allows that chlorotoxin, it allows the chlorotoxin into the cell. Now, the chlorotoxin have entered into the cell. Now, it will increase the CAMP levels and it will cause diarrhea. It increases the secretions in the JT. I'm not going in detail because this is all the microbiology part. But anyway, if you want to know, see, whenever you get cholera infection, what will happen, sir? It produces a toxin, chlorotoxin. This chlorotoxin enters into the cell with the help of Gm1 ganglioside. Now, when the chlorotoxin enters into the cell, what it will do? It increases the CAMP levels, MCQ. It increases the CAMP levels. CAMP means cyclic AMP, the secondary messenger. It's an example of secondary messenger. What happens if CAMP levels increases in the cells, especially in the GIT? This is all happening in the GIT. So what happens now in the GIT? More sodium and water is now getting secreted into the GIT. Now all this water, sodium, what will happen? Comes out in the form of stools, causes a diarrhea. Now my question for you, if you are really good, cholera means diarrhea, right? Now if I am the one who is suffering with cholera, Okay, now the diarrhea, the consistency of diarrhea, it looks like, okay, is it a pea soup diarrhea, or the consistency of the diarrhea is having a characteristic name? It looks like it watery diarrhea, but which water? Can you tell me, anyone, if you are really good? Rice watery, excellent guys. Okay, it's a rice watery diarrhea. The diarrhea because of the color is rice water. Okay, the rice watery diarrhea, you will have color. MCQ which I want you to know. As we are discussing about the topic, I have just said pea soup diarrhea. Pea soup diarrhea. Can you tell me in which GI infection you will have pea soup diarrhea? In which GI tract infection? Okay, if you have this infection to your GI tract, you will have pea soup diarrhea. Can you tell me? Pea soup diarrhea is seen with anyone? Come on, Amir, Bruce, Cypria, anyone? Pea soup diarrhea, just like a pea soup. Typhoid, guys. Okay, infection with typhoid, Salmonella typhi. Whenever you have typhoid, it causes pea soup diarrhea. Okay, okay. Now let's come back to our topic. We have done with the lipids. Lipids, all our business is done. Now after lipids, we have to discuss about the proteins. Now let's start the protein part. From here it's going to be very much easy. Lipids is the biggest thing. Now let's start with the proteins. The first important point you should never forget, proteins are the major component. Okay, they are the major component in the cell membrane. Now what are the different types of proteins which you should know for your exam? The types of proteins, mainly the proteins are classified into three types. Okay, the proteins are mainly classified into three types. The first type is a transmembrane protein. Transmembrane protein. Okay, transmembrane protein. The name itself is there, right? Transmembrane, throughout the membrane there is a protein. So that protein which is spanning, throughout the membrane is called as a transmembrane protein. Okay, and there are also certain proteins which are present only in the peripheral sites. So that type of proteins are called as the peripheral proteins. First, let me write it down here. Transmembrane protein is the first type. Second type is peripheral protein, and the third type of proteins are called as lipid anchored. I ask you to remember this lipid anchored protein. Okay, lipid anchored protein. Three important proteins. First of all, let's talk about the transmembrane proteins. Important points which you need to know. What exactly is a transmembrane protein? The protein which is spanning throughout the membrane. Throughout the membrane means it's passing through the membrane. Okay, entire membrane is passing. Now MCQs which will come in your exam is, they will ask you what are the examples. What are the examples of these transmembrane proteins? The examples are receptors. Okay, the receptors are examples of, okay, the receptors, especially G protein coupled receptor, GPCR. Tomorrow's class we will discuss this in detail, GPCR. For example, okay, let me show you here for your understanding. If this is the cell membrane, okay, this is the cell membrane, the bilipid layer. See, this is how the G protein coupled receptor is present. Okay, G protein coupled receptor. See, it's having 7 turns. It's passing through the membrane 7 times. Inside the cell membrane, it is bound with a protein called as a G protein. So it's called as a G protein coupled receptor. Okay. Is there any jerk? Okay. Let me repeat it again. Okay guys, what I am discussing here is the transmembrane proteins. So I am discussing about the transmembrane proteins. The example of a transmembrane protein which I want you to know is the receptors. Which receptor? G protein coupled receptor, sir. This G protein coupled receptor, it is passing through the entire cell membrane. Okay, next, the other examples are pumps. PUMPs, pumps. So direct question, pumps are examples of phospholipid, glycolipid, cholesterol, proteins. Proteins, which type of protein? Transmembrane protein, integral protein, peripheral protein, integral proteins, different, which type of protein? It's an example of transmembrane protein. Okay, Now, second thing, what exactly are these pumps? For example, can you give me any example of a pump? Pump means it's a channel which uses ATP. For example, let me show you here. See, imagine this is a pump. Okay, this is a cell. On the surface of cell, there is a pump. The name of this pump is sodium, potassium, ATPase. Sodium, potassium, ATPase. Now, whenever in your life you listen this word pump uses ATP. Pumps are all the time uses ATP. ATP means active transport, primary active transport. Okay. So, what exactly it is doing? It's a protein. It is allowing potassium into the cell, 2 potassium ions into the cell, and throws 3 sodium ions out of the cell. It's a channel. It's a protein. It is bringing 2 potassium into the cell and throwing 3 sodium out of the cell with the help of ATP, by using ATP. So these pumps are also an example of transmembrane proteins. They are present in the entire length of the cell membrane, completed, done. And all the ion channels. Okay, the second example completed. The third example is ion channels. Ion channels, sodium ion channel, chloride channel, potassium channel. The channels are also examples of the transmembrane proteins. Completed, done. Okay. Now, peripheral proteins. Here is a very important area you should know. Peripheral proteins means what? They are either present outside the cell membrane or they are present inside the cell membrane. If this is the cell membrane, see, they are either present outside or they are either present inside. Okay, these type of proteins are called as the peripheral proteins. Can you name me some important peripheral proteins which you know? 3 important proteins which you should know for your exams, guys. 3 important types of peripheral proteins are: first protein is spectrin, ankyrin, and dystrophin. Okay, spectrin, ankyrin, dystrophin, these are the peripheral proteins. Important points for the exam is, first let's talk about the spectrin. Okay, let me write it down here. Spectrin, and ankyrin. Now, the spectrin and ankyrin, where they are present? They are present especially in the RBC cell membranes. RBC means red blood cell, RBC cell membranes. Now, have you ever wondered why the RBC is something like this? Okay, biconcave in shape. Biconcave. Why the RBC is biconcave in shape? Why not the RBC simply round or spherical in shape? See, the RBC is biconcave in shape is because of these 2 important peripheral proteins, the spectrin and ankyrin. Now, in your exam, the biconcave shape of RBC is due to spectrin and ankyrin. For example, now if spectrin is defective, if there is any defect in the spectrin, or if there is any defect in the ankyrin, what will happen if there is any defect? Can you tell me, anyone? If there is defect in spectrin, it will cause spectrin for hereditary elliptocytosis. If there is any defect in spectrin, it causes hereditary elliptocytosis, not spherocytosis. Spectrin defect will cause hereditary elliptocytosis. RBCs will become elliptical. Now, defect in ankyrin, repeatedly as an MCQ, very important point. Ankyrin defect will cause hereditary, okay, hereditary spherocytosis. Okay, spherocytosis. This is the MCQ. Means RBCs are becoming more like a spear or round shape. Hereditary spherocytosis is due to defect in ankyrin. Very important. So 2 important peripheral proteins are completed. Ankyrin and spectrin are completed. Now let's talk about the third peripheral protein, which is dystrophin. So what you should know regarding dystrophin, sir? Very important point is dystrophin gene. Dystrophin is a protein. Dystrophin gene is the largest gene. Okay, it's the largest gene in humans. Okay, the largest gene ever found in the humans is the dystrophin gene. Now, whenever there is a transcription and translation of the dystrophin gene, you will be having a protein called as a dystrophin, and that dystrophin is especially present in the muscles, skeletal muscles. It's attached to the cell membrane. Important point is whenever you're having defect in this dystrophin, it will cause a disease called as, anyone? Can you tell me? It causes a disease called as a DMD. DMD. What exactly is DMD? Can you tell me? What exactly is DMD? Yes, excellent guys. It is Duchenne's muscular dystrophy. Duchenne's muscular dystrophy. Okay, so DMD means Duchenne's muscular dystrophy. Now, especially for the exam point of view, here I want you to know. Now, if I am the person who is having Duchenne's muscular dystrophy, do you think my skeletal muscles are going to work properly? My skeletal muscles are not going to work properly, guys, for sure. I'm going to have weakness in my skeletal muscles. Now, if I squat down, and if I want to just lift up, if I just want to stand up, can I do it just like a normal person? Definitely not. I will just bend forward. I will take the support of the ground, okay, and then I will try to stand up. So this is the sign which is seen in the person who is suffering with the Duchenne's muscular dystrophy. The sign is called as. Can anyone tell me? Okay, let me show you the image. See, this is the person. Okay, he's sitting down, and if I ask this person to stand up, see how he is standing. He's taking the support of his upper limbs also. So this sign, can anyone tell me? Sign name is called as the Grover sign. Grover sign. The Grover sign. Grover sign. Grover sign. So Grover sign is seen in DMD, Duchenne's muscular dystrophy. Done. Now after the peripheral proteins, the last important protein which I want you to know is the lipid anchored protein. Okay, lipid anchored protein. The lipid anchored protein name is GPI. GPI. The glycosyl phosphatidylcholine. Okay, let me write it down here. Glycosyl phosphatidylcholine. Okay, glycosyl phosphatidylcholine. GPI. GPI is a lipid anchored protein. I ask you to remember. Have you just seen? Have you remembered? See, this is the cell membrane. Usually in the cell membrane, who are there? Lipids are there. Phospholipids are there. This is a phospholipid. What is the name of this phospholipid? The name of the phospholipid is inocytal triphosphate, IP3. Okay, inocytal triphosphate. To this lipid, to this phospholipid, there is a protein attached. The name of this protein is GPI. Okay, this protein, the pink color protein is GPI, glycosyl phosphatidylcholine. Why? Because it is attached to the phosphatidylcholine triphosphate. Okay. Now this pink protein is attached to a lipid. Now, just by looking at this image, just tell me how many arms does it have? How many? How many hands does it have? There are two hands, right? Screen, it's come back. Yes, it came back. Now, how many hands does it have? Guys, shall we take a break of 2 minutes? We need to do some technical things. Shall we take a break of just 2 minutes? Is it okay? Okay, just 2 minutes break. క్లోజ్ చేస్తామనండి. Okay, guys, let's come back. What we are discussing? We are discussing about a lipid anchored protein, right? Lipid anchored protein, the protein which is attached to a lipid. Now, see, it's having two hands, right? See, this protein, it's having two hands. Now, with one hand, it just catches up a molecule called as a DAF. DAF. Guys, this is very, very clinical important. We are discussing a clinical, we are just trying to integrate it with the clinical cases. Okay, there is something called as a DAF and MIRL. Don't worry, I will explain to you. So what I am saying is, especially, see, okay, let me write it down here. Especially on the surface of your RBCs. Okay, imagine this is your RBC. On the surface of your RBCs, there is inocytal triphosphate IP3. Phospholipid is there. Now, to this phospholipid, who is attached to this phospholipid, there is a protein attached, lipid anchored protein. What is the name of this lipid anchored protein? GPI, glycosyl phosphatidyl inositol. How many hands does it have? Two hands. With the two hands, it's actually catching two molecules. What are the name of two molecules? DAF, decay accelerating factor. Okay, DAF means decay accelerating factor, and MIRL, membrane induced reactive lysis. DAF and MIRL, DAF, just you don't need to know the definitions. Just remember DAF and MIRL are present on the GPI. Now, why we are discussing all these things, sir? See, actually, this DAF and MIRL are very, very important. They are all the time present on your RBCs. They are very important in your blood. Okay, let me show you a blood vessel. See, this is your blood vessel, right? In your blood, there are proteins called as the complement proteins. Okay, there are proteins called as the complement proteins. They are totally related to the immune system. Now, right now, I put a bacteria here into your blood. Okay, this is not a sperm, this is a bacteria. Okay, take as a bacteria. This is a bacteria. This is a bad guy. Now, whenever a bad guy present in your blood, who activates? Complement proteins are activated. Now, what these complement proteins are do? At the end of the day, there are cascade of events. Lot of events, lot of chain reactions will occur. At the end of the day, there is production of a molecule called as MAC, MAC. Okay, membrane attack complex. Let me write it down here. MEMBRAN, membrane ATTAC, attack complex. So, when complement system is activated, who is going to be produced? Membrane attack complex is going to be produced. Now, what this membrane attack complex will do? Actually, membrane attack complex will attack the bacteria and will kill the bacteria. My question is, there is a probability, there is a chance that this membrane attack complex can also come and attack the RBCs. Why? Because RBCs are also cells. Now, whenever you get infection, does your RBCs are getting destroyed? No. Why? Because see, whenever there is an infection, whenever there is sepsis, or whenever you are having infection, the complement system is activated, membrane attack complex is produced, but this membrane attack complex is only going to kill the bacteria, not the RBC, because on the surface of RBC, there are DAF and MIRL, decay accelerating factor and membrane induced reactive lysis. These two substances will inactivate the complements, will inactivate the coming complements, so that there is no problem to the RBCs, there is no hemolysis. But for example, now I'm going to talk about a condition, pathological condition called as paroxysmal nocturnal hemoglobinuria. So what exactly is this condition? What exactly is the problem in paroxysmal nocturnal hemoglobinuria? It's simple, absence of GPI. The GPI is not there. Whenever there is no GPI, there is no DAF, there is no MIRL. So what happens now? See, okay, what happens now? The problem is going to be, the patient is going to suffer with hemoglobinuria. Whenever he goes to urine, his urine is going to be dark red in color. Hemoglobinuria. Don't confuse. You will think like, sir, why the urine is coming red in color? Paroxysmally, sometimes, especially in the night, nocturnal, sometimes in the night, whenever the patient goes to the urine is going to be especially dark red in color. Now, why this happens is because, see, whenever you sleep, what happens? Now, what kind of breathing do you have, guys, whenever you sleep? Okay, whenever you sleep, what kind of breathing pattern you have? You have a shallow breathing. Okay, you have shallow breathing, means you're not going to have deep inspiration. You are not going to do yoga, right? Especially when you are sleeping, you are going to have a very subtle respiration. Shallow respiration is going to be there. Whenever there is a shallow breathing, carbon dioxide will properly come out of your lungs. Proper washout of carbon dioxide is going to be there. No, carbon dioxide retention. Carbon dioxide is going to be present, retained in the body. What happens to this carbon dioxide is carbon dioxide will react with the water in the lungs. Now it forms H2CO3. Now this H2CO3 is going to split into HCO3 minus plus H plus. Now these protons are being these protons are being generated. The protons, H plus ions, are being generated in your body and in my body, especially when you are sleeping. So during sleep, what happens? Our blood is going to be more acidotic, more protons. Protons are acids. So our blood is going to be more acidic, especially when we sleep. Now, why we are discussing all this stuff, sir, is because now acidic pH, this acidic pH will activate complements. This acidic pH actually activate the complements in your body and in my body. Complements are activated. Complements are activated, especially when we are sleeping. Okay. Now this activated complements thing will now come to the RBC. Normal healthy person RBC. Now activated complements are coming to the RBC in a healthy person. What happens? DAF and MIRL are there. DAF and MIRL will inactivate the complements. But in a person who is suffering with PNH, paroxysmal nocturnal hemoglobinuria, GPI is not there, DAF is not there. So what happened? Now complements are not inactivated. Complements are directly coming and attacking the RBC. So now what happens to the RBC? RBCs are going to be ruptured. Hemolysis is happening. So who is going to come out of the RBC? Who is going to come out of the RBC? Hemoglobin will come out of the RBC, the pigment, red color pigment. This hemoglobin will go into the urine. So whenever this person goes to the urine, he is going to have dark color urine. So that's the reason why paroxysmal nocturnal means night hemoglobinuria. So this is how the pathophysiology occurs in the paroxysmal nocturnal. Okay, so what you need to know regarding paroxysmal nocturnal hemoglobinuria is simple things. Absence of GPI. Okay, GPI is absent. No DAF. DAF is not there. DAF is not there. MIRL, membrane induced reactive lysis are not there. Now repeatedly asked questions from, especially from the patho point of view. In the exam, they won't give you DAF. They won't give you DAF and they won't give you MIRL. Okay, instead of DAF and MIRL, they will give the name something like CD55. CD55 is other name for DAF. It's a CD marker, cluster designation marker. CD55 is DAF and MIRL is CD59. Cluster designation factor number 59 is MIRLS. So paroxysmal nocturnal hemoglobinuria is also completed. Now after this, what else we should know, sir? Especially from the membrane physiology, I want you to know some important points about the junctional complexes. We are about to end the important topic. Some important topic, especially this junctional complexes topic, will be integrated with the derma point of view. Especially from the dermatology, these questions are very, very important. Okay, so what exactly is this junctional complexes, sir? The first junctional complex which I want you to know is desmosomes. Desmosomes. Okay, see, name itself is a junction. Junction in between two cells. Two cells are having a junction. Okay, what exactly are these desmosomes? Oh, first of all, let me show you one cell, skin cell. This is one skin cell. The skin cell is something like this. Okay, this is one skin cell and this is the other skin cell. Now, my question for you, if you are really good, try to answer it. If you are a real medical student, just try to answer it. Okay, it's very hard for most of the students. See, every student knows, like, the cell in the lungs are called as alveolar cells. The cells in the liver are called as the hepatocytes. So what is the cell? What is that cell in the skin? Can anyone tell me what is the skin cell called as? Anyone, guys? Can you really tell me what is the skin cell called as? If you are really good, just tell me. Come on, Bruce, I want you to answer. Bruce, Frederick, Medico Levan, Saad, Amir. Anyone? What exactly is a skin cell called as? These are all epithelial cells we are having right here. The skin, which is made up of 5 different layers: stratum corneum, granulosum, lucidum, spinosum, basal layer. Now the skin cells are made up of what? Carniocytes? No. Dermacytes? No. Keratocytes. Okay, don't just invent a new thing. Okay, the skin cells are called as keratinocytes. Keratinocytes. Okay, now especially here I am talking about the cells, especially cells in the skin. Now see, this is the basement membrane. The basement membrane. On the basement membrane, cells are sitting. On the basement membrane, the cells are sitting. Now see, okay, let me show you in a very simple way. This is the basement membrane. These are the cells, guys. These are the cells. These are the keratinocytes. Keratinocytes are sitting like this. Now, one keratinocyte is attached with the other keratinocyte. Okay, one keratinocyte is attached with the other keratinocyte with the help of which proteins? With the help of proteins called as desmosomes. MCQ, desmosomes. Now, other question. Now, the same keratinocyte, it is sitting on the basement membrane, right? Yes, it is sitting on the basement membrane, and here also you need to have attachment. Now, what is this protein which is attaching a keratinocyte to the basement membrane? Can anyone tell me? This is hemidesmosome. Hemidesmosome. Hemidesmosomes. So, desmosomes and hemidesmosomes. Especially from the exam point of view, the questions repeatedly will come on desmosomes. Desmosomes. Now, you have to answer, guys. Desmosome is not a one single protein. Desmosome is again made up of different proteins. Desmosome is a complex. It's a junctional complex. Desmosome. Now, can you tell me what are the desmosomal proteins? Can you tell me what are the desmosomal proteins? MCQ, thermo point of view. Desmosomal proteins. Anyone? Desmoglin. Not connexin. Oh my god. Desmoglin. Desmocolin. Desmocolin. Desmoplakin and placoglobin. See, these are the desmosomal proteins. Desmoglin, desmocolin, desmoplakin and placoglobin. These are the four different types of desmosomal proteins. Now, important MCQ is, whenever you have damage, just think logically and tell me, whenever you have a damage to this desmosomal proteins. Normally, the cells are like this. Each and every cell is attached like this with the help of these desmosomes. See, desmosomes are attaching each and every cell. Whenever you have any problem, any defect with this desmosomes, what happens? Now, do you think cells are going to stay together? All the cells are going to stay together? No. What will happen? Now cells start to separate from each other. Our skin cells, now they will move from each other. So there is a space. Now there is going to be formation of a space or a lacuna between the keratinocytes. Now what will happen? Wherever you have empty spaces in your body, all those empty spaces are going to be filled with fluid. This is the concept. Now you are going to have what? Blister formation in your skin. Okay. Now see, whenever you have a damage, please concentrate. Whenever you have a damage in these desmosomes, what happen? Now these keratinocytes, they are going to separate and become round in shape. Now these separated keratinocytes are called as, can you tell me? Separated keratinocytes. Keratinocytes. Separate keratinocytes are called as anyone? Separated keratinocytes which are like not attaching to anyone. These separate keratinocytes are called as acantholytic cells. Acantholytic cells, very important MCQ. A separated keratinocyte which is not attaching to anything, which is not attaching to the other cells are called as the acantholytic cells, guys. Acantholytic cells. Okay. Now, what else we have to know for our exams? In exam, what kind of questions will be asked? Okay, in any kind of exam, these are the kind of questions will be asked. See, desmoglin, especially desmoglin 3, if you want to know in specific, the desmoglin, it's a desmosomal protein, helps in attachment of the keratinocytes. Desmoglin 3, if it is defective, it causes a disease, it causes a skin disorder, where you are going to have lumps, where you are going to have blisters on your skin. Not lump, where you are going to have blisters on your skin. Can you name me that disease? Desmoglin 3 defect will cause a disease called as pemphigus vulgaris. Okay. Pemphigus vulgaris. Okay, Pemphigus vulgaris is a blistering disorder. It's a blistering disorder due to damage in desmoglin 3. Another question is, see, whenever you have damage to or defective in desmoplakin or desmoplakin and plakoglobin, whenever they are defective, it causes a disease. It causes again a skin disorder where you are going to have again acantholytic cells. Can you tell me what is the disease? Anyone? If you know, if you are really good, especially if you are going to the exam, okay, especially if you are the guys who are watching, those guys who are going to the exam, Fmge exam or the Neet PG exam, can you tell me desmoplakin and plakoglobin are defective in which disease? Paraneoplastic pemphigus, guys. Paraneoplastic pemphigus. See, these are the direct single liner MCQs which are kept on coming in the exam. Plakoglobin, desmoplakin. Okay, plakoglobin, desmoplakin, whenever they are defective, it causes paraneoplastic pemphigus. Desmoglin 3 defective will causes pemphigus vulgaris. Pemphigus vulgaris. As you guys are very new to this medicine thing, okay, I hope, like, the information whatever I have, most of the students who are watching, they are especially from the, like, the first years and the second years. See, whenever you listen this vulgaris means most common. Whenever you listen the word vulgaris, it indicates most common. Pemphigus vulgaris means the form of pemphigus which is most common. Okay, there are other types like pemphigus foliaceus, other things. Okay, sir. Now after this, what else we have to know? Let's talk about some important points regarding the hemidesmosomes. Hemidesmosomes. I have already explained to you, this is the basement membrane. On the basement membrane, all these keratinocytes are sitting like this, and these keratinocytes are bound to the basement membrane with the help of hemidesmosomal proteins. Now, my question is, what are the examples of the hemidesmosomal proteins? Hemidesmosomal protein is not a one single protein. Okay, hemidesmosome is not a one single protein, it's also a junctional complex. Now, can you name me if you are really good? The hemidesmosomal proteins, can you tell me? Anyone? Come on, guys. Hemidesmosomal proteins. We are about to end. Come on, come on. We have to wrap it up as fast as possible. I want you to answer. I want you to be excellent. BPAG1. BPAG1. What is exactly is BPAG? Not needed. If you want to know, BPAG means bullous pemphigoid antigen 1. BPAG1, BPAG2, plectin, integrin. BPAG1, BPAG2, plectin, integrin, these are hemidesmosomal proteins. Now, what are desmosomal proteins? Desmosomal proteins are desmoglin, desmocholine, plakoglobin, and desmoplakin. These are desmosomal proteins. Desmosomal protein defective will cause pemphigus vulgaris, paraneoplastic pemphigus. Now, my question is, if you have any defect in hemidesmosomes, what happens? Now, you will have blister where? Within the epidermis or below the epidermis. Now, whenever you have any problem to hemidesmosomes, again cells will separate, again blister will form. But where now blister will form? So, blisters are going to form below the epidermis. Why? Because the hemidesmosomes are the ones which attach keratinocytes to base membrane. Now, whenever the cells separate, you are going to have a blister below the epidermis. Okay, so subepidermal bulla, the subepidermal blisters will form. Okay, now, okay, anyway, you will study this in later in the pathology also. Now, my question is, so whenever you have damage to BPAG1 and BPAG2, it causes a disease called as pemphigus? No, the disease name is called as bullous pemphigoid. Bullous pemphigoid. Repeatedly ask MCQ in your exams. Bullous pemphigoid is due to defect in hemidesmosomal proteins. Which hemidesmosomal proteins? BPAG1, BPAG2. Okay, and especially this BPAG2 is affected in most of the disorders, skin disorders, especially like linear IgA disease, linear IgA disease, and something called as herpes gestationalis. Okay, so in diseases like linear IgA disease and herpes gestationalis, the defective protein is hemidesmosomes. That too especially BPAG2. Okay. Now after this, what else you should know regarding the membrane physiology, especially regarding the junctional complexes? Very simple thing. Gap junctions. 2-3 important points regarding gap junctions. Gap junctions. I'm very sorry for my handwriting. Okay, I'm very bad with my handwriting. Okay, the gap junctions. The third important point is the gap junctions. What exactly are these gap junctions? Now let me show you the image. Simple image which I have downloaded. See here. These are the gap junctions. See, this is one cell. This is the other cell membrane. The one cell membrane, other cell membrane. Now, are you able to appreciate these gaps? Like, it's a made up of 6 proteins coming together and it's actually forming a gap which allows the movement of ions from one cell to other cells, which allows the movement of substances from one cell to the other cell. So these are known as gap junctions. Important MCQ which I want you to know for exam is these gap junctions are made out of a protein called as connexons. Okay, connexons or connexins. Connexons or connexins are the proteins which are forming gap junctions. Okay, let me write it down here. Gap junctions are made up of connexins. And MCQs in the exam which I have previously asked is, see this connexin 32. There is a protein called as connexin 32, C32, connexin 32. And there is something called as connexin 40, connexin 32 and connexin 40. Now, whenever you have any damage to these connexons, gap junctions, connexons mean gap junctions. Whenever you have damage to this connexin 32 or connexin 40, these persons will actually end up in a disease. Can you tell me? Connexin 32 mutation, connexin 32 defect will cause a disease called as Charcot-Marie tooth disease. Direct MCQ, Charcot-Marie tooth disease is due to a defect in connexin 32. Okay, what exactly is Charcot-Marie tooth disease? We will study in your pathology. Right now, this is not a class of pathology. Okay, whenever I teach you pathology, then we will discuss. Now, connexin 40 defect will cause idiopathic atrial fibrillation. Okay, idiopathic atrial fibrillation. So, idiopathic atrial fibrillation is due to defect in connexin 40. Okay, now after this, last junctional complex which we are discussing is tight junction. Tight junction. Now these junctions are very tight, attaching the cell very strongly. Okay, very strongly. These are attaching the two different cells very strongly. Now these tight junctions, they will allow only certain substances selectively permeable. For example, if this is one cell. Okay, this is the other cell. Now what do we have is a tight junction. Tight junction means just like desmosomes, these tight junctions are also attaching two cells together. Okay, now these tight junctions, the MCQ which was asked is proteins. Now tight junctions are made up of which proteins? Just like desmosomal proteins, semi-desmosomal proteins. Gap junctions are made up of connexins. Just like that, tight junctions are also proteins which are helping in attachment of the two cells. Now these tight junction proteins are, can you tell me, there are 3 important tight junction proteins. Anyone? Can you tell me? Anyone? Excellent, Bruce. What is that? Clues. What is that? Claudins, guys. Claudins, excellent forever, excellent Bruce. Claudins, occludins, and junctional adhesion molecules. So claudins, occludins, and junctional adhesion molecules. All these are the examples of tight junction. Tight junctions attach the cells, two different cells, very tightly. If you want to know where exactly tight junctions are present in your body, tight junctions are present in, not in the skin. In the skin, you are having desmosomes. Okay, tight junctions are very tight. They are present in blood-brain barrier. Tight junctions are present in blood-brain barrier, guys. Very important MCQ. Okay, tight junctions are present in blood-brain barrier. Okay, with this we have completed the basic points which you need to know regarding the cell membrane. Okay, this is the first class of the general physiology. We have started with the cell membrane, just simple topic cell membrane. I tried my best to give you maximum, okay, which is needed for your exam. Hope the class is helpful. If you have any questions, you can all the time personally message me, okay, or you can also just ask your question in the comment section. Okay, after the video you will be having the comment side, you can post your questions. I will try my maximum to give you the answer. Tomorrow let's meet up with a new topic. Okay guys, thank you. Yeah, lining of the gut also, yes, excellent. Okay, lining of the gut, in the lining of the gut, blood brain barrier, blood testicular barrier, ovarian barrier, in all these places you will be having this tight junctions. But exam point of view, what you need to know is tight junctions are made up of which proteins? Codins, occludins and junctional adhesion molecules. Okay, junctional adhesion molecules, which are also called as the jam, junctional adhesion molecule, JAM. Okay, thank you guys, bye bye, see you tomorrow.